There we go. Okay. So hello, and good morning, everyone, or evening, depending where you are at. My name is Michelle Hermiston. I am currently the chair of the Langerhans Cell Histiocytosis Steering Committee for the Histiocyte Society, and I want to warmly welcome all of you to our webinar, Potential New Frontiers in Therapy for Langerhans Cell Histiocytosis. As many of you know, the Histiocyte Society is a nonprofit organization of over 200 physicians and scientists from around the globe who are committed to advancing knowledge and improving outcomes for patients with histiocytic disorders. We focus on clinical and basic research as well as education, and this session is one in a series of webinars from the Histiocyte Society and is being hosted by the LCH Committee. This is definitely a team effort, and I do want to highlight the committee members who helped to develop the webinar today. And as many of you will note, there's also some people who terms are ending, and we're always excited to involve new people, especially younger people who maybe haven't gotten involved in this society. So if you are interested in potentially serving on the committee, please do reach out to us. I'd be happy to chat. A couple of housekeeping points. We've muted all of the participants, so please submit your comments via the chat feature in your webinar panel. It will be steamed by the staff and the panelists, the chat is in the controls at the bottom. It should look like this, and after clicking, the chat window will appear on the right, and you can type in your message and then press enter or return to send it. Please put questions for the speakers in the Q&A box. These will be addressed at the end, so please indicate who the question is for. Please put questions for the staff in the chat box, and Kathy will help with those. And then like a question in the Q&A box, if your question has already been entered by somebody else, and that will pop it up so that we can prioritize that. It is likely due to time constraints, we may not be able to answer all questions, but I'm sure that the speakers would be happy to email and answer questions afterwards. Finally, the webinar is being recorded and will be sent to all registrants. The Histiocyte Society members will also be able to find the video in the members site in our website. And before we go to the presentations, I want to take just one quick moment to thank Kathy, who coordinated all of this and as always does just such a wonderful job supporting all of us. So a quick virtual round of applause for Kathy, and then we will get started. Today we have 3 presentations that were selected by the committee. 2 are top ranked LCH abstracts that were submitted for the annual meeting. The role of BRAF mutated hematopoietic cells in patients with Langerhans cell histiocytosis from Carolyn Hunter's group, and Sebastian Elder will be speaking and presenting this work today. They'll be followed by a clinical study of MAP2K1 mutated LCH in children from the group in one of the groups from China, and Dr. Jun Wang will be presenting that work. And then finally, one of the things we've been discussing in the committee is thinking about the microenvironment and the place that these cells live in, and what is actually happening from a pathophysiology standpoint, and could this in LCH, and could this lead to new therapies? And so the final talk will be from Miriam Murad and Carl Allen's group, who had a very nice paper in Nature Medicine earlier this year, BRAFV600E Induced Senescence Drives Langerhans Cell Histiocytosis Pathophysiology. Camille Bingenwald and Matthias Wolk will be presenting that work. So I will stop sharing and hand it over to Sebastian for the first presentation. Thank you very much.
Hello, can you hear me and see my slides?
Perfect.
Yeah, okay. So hello and good afternoon from Austria. My name is Sebastian Eder. I'm a pediatric resident and a member of Professor Carolina Hutter's LCH biology group at the Children's Cancer Research Institute in Vienna. And thank you for the opportunity to present our data in this webinar about BRAF mutated hematopoietic cells in LCH patients. As you all know, the most common mutation LCH is the BRAFV600E mutation with about 60% of the cases. BRAF is a common oncogene associated with the activation of the ERK pathway. And the BRAF allele load was shown to correlate positively with the disease activity score at the time point of diagnosis. And on the right, we see that in patients with a low disease activity score, only 20% had a positive BRAF mutation, whereas all patients with a disease activity score of 7 or higher had the BRAF mutation. It was shown by different groups that BRAF allele burden could be decreased and even eradicated with chemotherapy. So the BRAF level in peripheral blood is a promising biomarker in patients with this mutation. Besides chemotherapy, there's also BRAF inhibitors like vemurafenib. Vemurafenib is well tolerated and effective. On the right, we see that the disease activity score at diagnosis could be well reduced through RAF inhibition in risk organ positive patients as well as in risk organ negative patients. And there is no development of resistance against the medication, which makes BRAF inhibitors an attractive therapy option. But all patients relapse after discontinuation, and this is because vemurafenib does not eliminate BRAF mutated cells. This could be shown by different study groups in allele analysis of the whole blood, as well as circulating cell-free DNA. And during the therapy with RAF inhibition, the BRAF levels always stayed elevated. So I want to present our data of a patient where we combined vemurafenib and chemotherapy. At the time of diagnosis, the patient was 2 years old with a multisystemic LCH with risk organ positivity, in his case, the bone marrow. And stratum 1 therapy with prednisone and vinblastine was started and did not lead to remission. So the patient was eligible for stratum 3 therapy, but was clinically in such bad condition that it could not be started. So we started therapy with vemurafenib 2 times a day, 10 milligrams per kilogram. And the clinical condition improved within a short time. In the top graph, you can see the body temperature. The fever went away, and in the graph below, the CRP levels normalized. Also, the pancytopenia normalized. And so, like a switch, the disease activity score around day 40 got better with vemurafenib. So as I said, it's like a switch. The clinical improvement in the patient was directly linked to the administration of vemurafenib. We also kept track of the BRAF levels, and as you see in the graph below. And interestingly, during vemurafenib therapy, the BRAF levels rose. So it is very important that the BRAFV600E allele load, in this case, did not correlate with the disease activity score during vemurafenib therapy. After the patient got better, we could treat him with stratum 3, so with cytarabine and cladribine, according to the LCH4 protocol. And during the chemotherapy cycles, vemurafenib was paused, and you see during these pauses of vemurafenib, the fever got worse and the CRP rose again, and also the clinical, the rest of the clinic got worse, but with starting again, it like a switch, it got better. Continuation therapy, vemurafenib was lowered in 2 steps and finally discontinued. And as you see below, the BRAFV600E levels stayed negative, and they stayed the therapy was completed according to the LCH4 protocol, and the diagnosis is now 3 years away, and the patient is BRAF negative ever since. So we wanted to analyze why vemurafenib is associated with such a direct impact on the clinic, and we wanted to know which cells had the BRAF mutation. And we looked for BRAF alleles in the whole blood sample at diagnosis and during therapy, and detected mutated cells in all hematopoietic cells, mostly in the myeloid cells, so mostly in monocytes and dendritic cells, but also in natural killer cells and granulocytes. So for further analysis, we performed single-cell RNA sequencing from the peripheral blood as well as the bone marrow at time points of diagnosis and during vemurafenib therapy. And we analyzed the transcriptome of about 5,000 to 6,000 cells per sample, clustered them accordingly to cell type, and analyzed the differences between diagnosis and therapy. And we found that the cytokines were less expressed during vemurafenib therapy compared to diagnosis. Here on the left, so the green ones is the therapy, and the red ones is during diagnosis. And for example, I'm showing here interleukin-1 beta and CXCL8, which were associated with LCH previously. Also, we performed the gene ontology pathway analysis and could show that a negative enrichment of immune and cytokine pathways during vemurafenib therapy, mostly in monocytes, but also in hematopoietic stem cells. So we think that this fast treatment response of vemurafenib could be due to the inhibition of the ERK signaling in these non-LCH cells. Although this is just one patient, we think the analysis is very comprehensive, and concluding, we think that the BRAF mutated cells, other than CD1A, CD207 positive cells, contribute directly to the clinical picture of LCH, and that RAF inhibition inhibits inflammation and leads to rapid improvement of the clinical picture, but does not eradicate the mutated clone. But a combination with intensive chemotherapy and the supportive therapy of vemurafenib could eradicate it. So our idea from this work is to reframe LCH, that risk organ positivity is not defined through the infiltration of liver, spleen, or the bone marrow, but clinically, and we should definitely have a closer look at the non-CD1A Langerhans positive, but the BRAF mutated cells, because we think the double positive LCH cells are just the tip of the iceberg. Thank you.
Thank you, Sebastian. That was fantastic. I think it nicely highlights one of the issues that we face when we're treating these children, and that vemurafenib and MAP kinase pathway inhibitors can be very helpful for these patients, but the exit strategy is really challenging. And I think this highlights something that would be great to actually test in a clinical trial. There are a few questions. First, From Kim Nichols, how often do you see a rise in BRAF levels in patients with LCH who are on vemurafenib?
Sorry, I was on mute. We have presented the rising levels of vemurafenib, of BRAF during vemurafenib before. Ila Schwentner from our lab had this publication, but other than that, in this patient, and yes.
Okay, great. And Core also had a few questions. The DAS is also dependent on transfusion frequency of both red blood cells and platelets. Therefore, even with no LCH activity, with the DAS will go up after 2 CDA Ara-C courses. Did you compensate for that?
I'm sorry,
I think you can read the questions. If you click on the
Ah,
Q&A box, they'll show up for you, and that might be easier.
Yes, and also I have to say that the disease activity score during stratum 1 is high and gets low during vemurafenib, but the rises in stratum 3, this is of course biased through the chemotherapy as well, because the cytopenia will result from the chemotherapy.
Great. And then Core was also curious, did you look at the RNA profiles of the 14 LCH cell subsets during inhibitor treatment?
Yes, but we had in the peripheral blood and the bone marrow, we had no double positive cells, and we did not have samples from the bone lesions. Okay.
And Millen had a nice comment in the chat. Those findings recapitulate through comprehensive test and observation we have done many years ago under the microscope, namely bone marrow patients. There's lots of questions going. Bone marrow of patients with severe disease is infiltrated by ordinary macrophages, but not by LCH cells. Ronald Go had a question that I also had. Do you know of any preclinical data showing synergistic activity between chemo and BRAF inhibitor? And I think a corollary to that, I was curious if there are specific agents that might synergize better with vemurafenib versus others.
Mm-hmm. No, I can't. I can't give an informed comment on that. Yeah,
I think it highlights, and this is why the LCH committee was excited about this work, potential directions in terms of clinical trials, as well as I think some much needed bench-based preclinical research. And then just two last comments and we'll move on to the next one. Thank you, Jennifer, for the pathology perspective. The ordinary macrophages in these patients are actually BRAF positive macrophages in the bone marrow. And it sounds like ECHO is collecting some of this data. So great. So thank you for a wonderful presentation, Sebastian. If you can stop sharing, we'll move on to the next presentation. Is Dr. Wang on, Kathy? Yes, awesome, great. Okay, so the next presentation is clinical study of MAP2K1 mutated Langerhans cell histiocytosis in children, and Dr. Wang will be presenting this work. Welcome, and go ahead and share your slides, please.
Hello, can you hear me?
Perfect, yep. I am not seeing your slides yet, though.
Can you hear it?
I can't hear
Can
you.
you see my PPT?
I am not seeing your PowerPoint.
Oh, sorry. Wait a moment.
No worries. While we're waiting, Sebastian, Astrid had a nice question in the chat and or in the Q&A, and maybe you can answer that. And I'm not seeing slides yet, but I am seeing that you're sharing your screen.
Is that okay?
I'm still not seeing slides. It says you have started screen sharing, but I'm only seeing black. I don't know if anybody else is seeing the same.
Can you see?
Nope, it's still black. And it, do you have two screens, and can you move your PowerPoint to the other screen? Was one suggestion someone had.
Or you can also email them to me at Secretariat, and I can pull them up as well.
Oh, sorry.
Okay. How about if you email them to Kathy at Secretariat, and you can email them to me, and I could also pull them up, and we'll move on to the last presentation, and then we'll be ready to come back to this. So the final presentation was a very beautiful paper from Miriam Murad and Carl Allen's group that was in Nature Medicine earlier this year. BRAFV600E induced senescence drives Langerhans cell histiocytosis pathophysiology. And we have Camille Bigemold and Matthias Wilk from here to present that work. So I'll let you guys go, and then we'll swing back to Dr. Wang's talk.
Okay, so do you hear me well?
Perfect.
Okay.
And we see the slides.
So,
excellent.
okay, good morning or good afternoon, depending on where you are. So I am Camille Bigenwald, and I was a former postdoc in the laboratory of Dr. Miriam Murad. And so this work was published a bit earlier in the year. And so it was made with Miriam Murad, of course, and also in close collaboration with Carl Allen and Jennifer Pikarsik. And of course, Matthias Wilk, who is also who is currently in Dr. Miriam Murad laboratory. So I will try to summarize our data. So as you all know, BRAFV600E mutation is expressed in more than half of LCH lesions. Next slide. And so because of the presence of this mutation, vemurafenib is currently used for treating LCH or Erdheim-Chester disease patients. And this treatment is extremely efficient. And we can also use cobimetinib in case of another mutation than BRAFV600E. And this treatment is also extremely efficient. But as Sebastian Eder told us in his previous presentation, although this treatment is efficient, these treatments cannot eradicate the BRAFV600E mutated clone. Moreover, these treatments can lead to severe toxicities, and it was demonstrated in a paper published in Blood a few years ago by Julien Roche team that when you stop the BRAF inhibitor, the disease relapses in about 80% of cases. So we have a real problem of eradicating in an efficient way the BRAFV600E clone. So as you also know, the BRAFV600E mutation inside LCH lesions are mostly found in Langerhans positive cells, but not only. But for example, not in T cells, as Sebastian Eder showed us also. And so basically our initial question, so next slide. We also know that the BRAFV600E mutation can be found in hematopoietic progenitors at a very low level, so usually less than 2% of the CD34 positive compartment are mutated. And so based on this data that you all know, our initial question was to try to understand how does a BRAFV600E positive hematopoietic progenitor give rise to mutated mononuclear phagocyte. And so next slide, to try to answer this question, we established different models. So first, a new mouse model that I will talk about a little bit later. Next slide. Then we also used cord blood cells that we transduced with a lentiviral vector expressing BRAFV600E coupled to a GFP cassette. And finally, we also got tissue samples, most notably bone marrow samples and FFP slides from brain of LCH neurodegenerative patients. So first, what I think is important in our data is that we were able to generate a new mouse model of Langerhans cell histiocytosis by inducing the BRAFV600E mutation extremely early in the hematopoiesis, so in hematopoietic stem cells that express SCL. So this is the promoter that we used. Okay, so just by inducing this mutation, the mice develop a LCH-like disease. So basically, next slide, they developed organomegaly, they developed a bone marrow aplasia, and we could observe a granuloma in the bones, and we could also observe an accumulation of BRAF of CD207 positive cells in all the organs that we examined. So the LCH mouse model that we observe using this mouse model is like a high risk, is a high risk multi-system LCH model, and you have to take into account that the percentage of mutated progenitors is about 30%. Okay, so it's much more than in humans. And we were also able to get humanized mice developing LCH by transplanting the cord blood cell transduced by our lentivirus vector. But these animals developed a very mild disease characterized by an accumulation of CD207 and CD1A positive cells in organs. So next slide, yes. Sorry, because we are doing the PowerPoint. We are two, Matthias and I, so it's a bit chaotic. I'm very sorry. Then, so as I told you, the percentage of mutated progenitors is 30%. Okay, and when, interestingly, what we observed in our LCH-like mice is that the hematopoietic stem cells mutated with BRAFV600E, they were not able to proliferate. As you can see here, there is a dramatic drop of hematopoietic stem cells in red, and they were more prone to differentiate into the mononuclear phagocytes, so that means into the granulocyte macrophage progenitors, and this was associated with an expansion of monocytes, macrophage, and dendritic cells in the bone marrow of our LCH mice. And we could confirm also that by doing transcriptional analysis. So basically, the mutated hematoprogenitors from our mice showed hyperexpression of genes associated with the macrophage and dendritic cell differentiation. And we also observed the same in human LCH bone marrow. Okay, and so what was interesting also in our model is that the cells that were actually not mutated for BRAF, but in close contact to BRAF/V600E mutated hematopoietic progenitors also had a tendency to differentiate more toward the mononuclear phagocyte. And actually, this was partially due to the fact that the mutated progenitors are secreting, have a particular secretome that are able to, that drive the differentiation into mononuclear phagocytes. So the capacity of the BRAF/V600E hematopoietic progenitors to differentiate to mononuclear phagocyte is driven by cell intrinsic and extrinsic cues. Okay, so then important, I think, in our data. So as I showed you a bit earlier, in our LCH mice, we did not see a hyperproliferation of mutated hematopoietic stem cells. So this intrigued us a lot, and actually, we started to think about the SENESCENCE program, which was something very very dark for me. And so I don't know if you are familiar with this physiological mechanisms, but SENESCENCE described like about 20 years ago, a bit less, and mostly in human Navy lesions. And so basically cells that undergo high stress, like an oncogenic stress, stop to proliferate, but they are still highly metabolic. They secrete a lot of cytokines, and they express anti-apoptotic protein. So next slide. So to say that a cell is senescent, there are many characteristics that are widely used in the senescence field. So first, the cells, of course, do not proliferate, and then they have to show canonical markers of senescence, such as high senescence-associated beta-galactosidase activity, high CDKN2 expression, which is a cell cycle gene, and they also have a senescence-associated secretory phenotype. So since it's a very blurry concept, there is some mouse model that have been generated, like the ink attack mouse model, to confirm that cells are senescent. Okay, so to confirm our hypothesis that the BRAF mutated cells are senescent, we indeed observed that there was no proliferation of mutated cells in the bone marrow and in the periphery. And so basically to do that, we used an experiment which is called bone marrow transplantation chimera assay. And we could observe that the BRAF mutated hematoprogenitors had a disadvantage proliferative capacity compared to wild type counterparts. Okay, we observed a low Ki67 positivity in tissue. Next slide. We also observed that in the bone marrow and in tissue, a high senescence-associated beta-galactosidase activity, and we also noticed a strong expression of CDKN2A gene. We also observed as well in the bone marrow and in tissue of our LCH mice a senescence-associated secretory phenotype. So this is which means a high secretion of IL-1, IL-6, and matrix metalloproteinase by the senescent cells. So in human lesions, we also observed that the LCH cells that are CD207 positives do not overexpress Ki67, conversely, for example, to lung cancer lesions. And using transcriptional analysis, we also observed that there is a high expression of CDKN2A exclusively in LCH cells compared to healthy monocytes or healthy T cells or healthy Langerhans cells. Okay, the fact that the LCH cells do not proliferate is actually not new, as it has been shown by previous groups. So in this slide, these magnificent stainings were made by Jennifer Pikarsik. And so this is, these slides are coming from a brain from a neurodegenerative LCH patients that also had circulating BRAFV600E cells. And you can see an infiltration of enlarged plump cells expressing CD1, 63, CD33, CD14, so myeloid markers. They were also expressing BRAFV600E, and they were also expressing CDKN2A. Of course, it's only one patient, but it's very interesting to notice that we observe this accumulation of myeloid cells overexpressing CDKN2A in the brain of LCH neurodegenerative patient. Next slide. This is also another experiment confirming the high senescence-associated beta-galactosidase activity specifically in CD1A positive cells in LCH lesions, conversely to healthy T cells coming from LCH lesions, or comparing to healthy Langerhans cells. And so basically, using our LCH mouse model, we could show that the LCH cells express all the canonical markers of senescence. These markers are widely used in the senescence field. Of course, they are not sufficient to perfectly confirm that the role of senescence is primordial in the pathophysiology of Langerhans histiocytosis. So this is why we had to generate a new mouse model. So basically in this new mouse model, we crossed our LCH mouse with ink attack mice. So in this very difficult mouse model, basically the senescence cells, the ones that are overexpressing CDKN2A, become GFP, so we can detect them and we can specifically delete them using a drug that is called AP. Okay, so we confirm using this new mouse model that there was an accumulation of GFP positive cells in the LCH mice. So the GFP positive cells are the senescent cells. And among these senescent cells, most of them were mononuclear phagocytes. Next slide. So this confirmed that indeed there is a senescence program in LCH. And then we decided to specifically delete the senescent cells using the AP drug. So it worked. As you can see, the GFP positive cells are deleted. And this led to a better prognosis of the mice. That means that their liver was, the mice presented less hepatomegaly and there was less immune infiltration. Next slide. So this is important to, this previous experiment is extremely important to confirm that indeed senescence is critical in LCH physiopathology, because when you delete senescent cells, the disease improves. And so we also saw in the senescence field, there are more and more drugs that are being developed to try to specifically delete senescence. And this is very difficult. And so far, the drugs that are being developed are mostly drugs targeting anti-apoptotic proteins, such as Bcl-xl inhibitor. So basically, we used ABT263, Bcl-xl inhibitor. So this is considered as a senolytic drug, but of course, it's not specific at all. And when we treated our LCH mice, we were able to specifically eliminate the cells that carry the BRAFV600E mutation. But this experiment was extremely tricky because ABT263 is extremely toxic in mice. And I wanted to show you a previous experiment that was made in Carl Allen's lab with Rikia Chakraborty. And here they got LCH lesions that they treated with ABT263, and they could observe an apoptosis of LCH cells. Next slide. So the take-home messages of our data is that BRAFV600E in LCH is probably occurring at a very immature level in the hematopoiesis. The cells undergo a huge stress and DNA damage response, and so they start to acquire a senescence program that is characterized by a lack of proliferation, a high metabolic activity, and a senescence-associated secretory phenotype. They differentiate into mononuclear phagocytes, and this is facilitated by the senescence-associated secretory phenotype. And in tissue, these senescent cells are resistant to apoptosis because of the increase of anti-apoptotic proteins. They do not proliferate, but they recruit a lot of immune cells thanks to this, because of the senescence-associated secretory phenotype, which lead to granuloma. And so I wanted to tell you, so next slide. So, so this senescence hypothesis has been confirmed by another Italian group with a very nice paper published in Nature Communication this summer. So we are not the only one to find this data. And so to summarize, what I wanted to tell you is that so LCH lesions show typical features of senescence in mice, but also in humans. Senescence might explain the very low fraction of mutated progenitors found in the bone marrow. So indeed, they have a proliferative disadvantage, and this is why we observe a solo mutated fraction in senescence compared to leukemia. And so this data also paves the way to a new kind of treatment, which are senolytics that could potentially eliminate the BRAFV600E clone that so far cannot be eliminated by vemurafenib and cobimetinib. So I really hope that senolytics, and most notably Bcl inhibitors, I don't know which one yet, could help to eradicate the clone. And finally, I wanted to thank, of course, all the laboratory of Myriam Merad and, of course, Myriam Merad, who is a great mentor, Jessica Leberichel and Matthias Wilk, and outside of Mount Sinai, Carl Allen, Rikia Chakraborty, and Jennifer Pikarsik.
Thank you so much, Camille. That was wonderful and really interesting, and I think gives some potential insights into some of the clinical findings in potential synergy issues that Sebastian was highlighting in his talk. We are limited on time, so I think if you can answer the questions, there's one in the Q&A from Astrid, and Michael has a couple of great questions in the chat. We'll move on to our last talk, which we were able to solve the problem, hopefully, via email. Are you able to see Dr. Wang's slides?
Yes. we can see
Awesome.
this.
Let me put it in presenter mode, and just let me know when you want to advance, and the floor is yours.
Hello, can you hear me?
Yes.
Oh, I'm sorry to have kept you waiting so long.
No worries.
Good morning and good afternoon. I'm honored to give the opportunity to share our study here. The topic of my study is genetic landscape and its prognostic significance in children with Langerhans cell histiocytosis. Several recent studies show that LCH cells originate from myeloid dendritic cells. Almost all LCH patients have diverse genetic alterations in MAPK pathways. About half of LCH patients carry BRAFV600E mutation. However, the prognostic impact of these mutations remains unknown. This slide was aimed to identify the LCH molecular landscape in childhood LCH biopsy using NGS. The prognostic significance of different gene mutations. Next slide, thank you. A total of 346 children with LCH were admitted in our hospital from January 2018 to October 2020. Of these patients, 223 patients with available biopsy samples for targeted sequencing were included in this study. Bcl cells were used as a paired germline control. 192 patients received systematic chemotherapy. 20 LCH patients were directly given targeted therapy because of very young age or severe disease. 11 patients with single-site involvement were observed without treatment. Next slide, thank you. This slide is just going to show the workflow of the experiment. Firstly, we take the lesion tissues for targeted sequencing of the customized gene panel. Then we detected the cell-free cell BRAFV600E in plasma by digital PCR. Thirdly, we identified the function of the mutants. Next slide. This is a customized gene panel, including 239 genes. Next slide. Next slide, thank you. This slide shows somatic mutations identified in LCH patients. BRAF mutations were mainly found in exon 12 and 15. V600E mutation was detected in 15.6% patients. In BRAF V600E negative patients, V600D mutation was detected in 8 patients. Among others, this BRAF deletion, BRAF 486 to T491 deletion in SK, was also common and detected in 7 patients. 17% of patients carried MAP2K1 mutations, mostly in exons 2 and 3, which encodes the autoregulatory domain and the catalytic core. The most common mutation in MAP2K1 was Q158 to E16 deletion, which was detected in 12 patients. Next slide. Thank you. This slide shows the proportions of mutations in pediatric LCH. We can notice that 8 patients carried 2 mutations in either 1 or 2 genes. 2 patients carried dual mutations in 1 gene with identical variant allele frequencies, suggesting that both mutations were on the same alleles. Other cases were detected with different VF, suggesting existence of LCH cell subclones. Next slide. Thank you. The FNBP1 BRAF fusion gene was identified in a 3-month boy with MSRO negative LCH. As shown in this figure A, the results showed 2 fusion transcripts. We further analyzed the fusion gene content exon 1 to 12 of FNBP1 and exons 8 to 18 of BRAF. The fusion retained the protein kinase domain of BRAF and could activate ERK phosphorylation. 2 novel mutations, MAP3K10, A17T, and R823C, were identified in 2 children with multi-born involvement. Overexpression of MAP3K10, A17T, and R823C could increase ERK phosphorylation, suggesting the potential role in LCH pathogenesis. Next slide, thank you. We further explored the functions of FNBP1 BRAF fusion gene and MAP3K10 mutations in cell proliferation and senescence. The 2 MAP3K10 mutations increase the cell growth comparable with BRAFV600E, while FNBP1 BRAF fusion decreased the cell growth compared to the control. Senescence markers CDKN2A and CDKN2B were highly expressed in cell overexpressing BRAFV600E, FNBP1 BRAF fusion, and the 2 MAP3K10 mutants. Their function of ERK phosphorylation was inhibited by trametinib. Next slide, thank you. We further analyzed the prognosis of patients who received chemotherapy. Patients were divided into 5 genetic groups according to mutations detected in this study. There was no significant difference in 3-year PFS among the 5 groups. In a multivariate analysis, age at diagnosis, involvement of skin or RO were independent prognostic factors. Next slide, thank you. Our previous study has shown that BRAFV600E in cell-free DNA was more closely associated with important clinical characteristics, treatment response at week 6, and prognostic than that in lesion tissue. Therefore, we divided patients with BRAF600E mutation into 2 groups, cell-free BRAFV600E positive group and negative group. As expected, there was significant difference in 3-year PFS among the 6 groups, with the lowest PFS in those with cell-free BRAFV600E positive group. Next slide, thank you. In conclusion, somatic mutations in MAPK pathway were identified in most of LCH patients. Patients with positive cell-free BRAFV600E mutation has the worst prognosis. The genotype may contribute to stratification and precise treatment for pediatric LCH patients. I would like to thank patients, their families, and our colleagues. Thanks for your patience. Thank you.
Wonderful. That was wonderful, Dr. Wang. Do we have any, I think we have time for maybe 1 or 2 questions. I think one of the advantages you have is just such a large number of patients. And so I think this is very important information because of that. One question I have is, given that there now seems to be groups that you can risk stratify, what are your thoughts regarding potential drugs to test or clinical trial design for the patients who seem to do worse?
Sorry, I don't quite follow you. Would you repeat it
Yeah,
again?
no, maybe we can ask it this way. So Core actually just put a question in, and if you click on the Q&A, you can also read it. There are two questions. One, why do you suggest that you can stratify if you don't see a clinical difference between different mutations? And Kim asked, was I wrong? But did you use a multi-gene panel? Were there other recurrent somatic mutations identified?
Our gene panel included in MAPK, PI3K, senescence, EMT, GST, and other cancer-associated pathways. We used the gene panel. The patient in our study received systematic chemotherapy.
Great. So I think we are actually at the hour. I want to thank all of the speakers for really interesting and thought-provoking presentations. Again, I want to thank Kathy for her help organizing or doing all of the work organizing this, and the LCH committee for helping to develop the program. I wish everybody a wonderful holiday, and please stay safe, and thank you very much.